Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
The variety of life on Earth is enormous. And much of it we can't even see. Dr. Carl Woese, with his research that used RNA comparison, gave us a different tree of life, one that includes a different perspective on microbes. We learned there are ancient microorganisms at the base of the tree, like the boiling mud-dwelling Thermatoga. We learned that we are all related, though the diversity of microbial life far exceeds the diversity of all other species of life put together. The diversity of microbes and the places in which they live is truly astonishing. Look in virtually any environment on Earth, even one as inhospitable as this, and you'll find microbes living there. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll discover how microbes came to be so diverse and how those differences are driven by their metabolic variety. The changing environments of the planet and the corresponding microbial genetic shuffling through eons of time have led to enormous differences in cell structure, as well as even greater diversity in metabolic capacities. Each unique metabolism allows an organism to exploit the resources of a particular environment. Though we all share the same chemical code of life, DNA, all life forms also differ genetically. Whoa, well these are new. You know, these skates are kind of like a mutation. Some organisms experience mutations that may or may not be beneficial depending on the environment. I can tell you that these skates don't exactly give me an edge in this environment. But if a mutation is selected for, that is, if the environment changes to give the mutated organism a selective advantage, like this, well then the mutation not only survives, but eventually spreads. This is the basis for the diversity of life. Different mutations in different environments give rise to different successful strategies for survival. Often, enough of the old conditions remain the same, and some members of the previous community persist. Perhaps in a more restricted scope. So, as environments change, new niches become available, and organisms adapt to fill them. Because of the niches they inhabit, their strategies for survival, their way of getting and using energy, their metabolisms, differ. Metabolic diversity follows from genetic diversity. But just what kind of microbial diversity are we talking about? Well, I'm here on the campus of Michigan State University to talk to Dr. Tom Schmidt, an assistant professor of microbiology who specializes in the study of microbial diversity. Dr. Schmidt, Jim Leinfelder.
Hello, Jim.
I think most of us have a general sense of what diversity means, but in the context of microbiology, what does diversity mean?
Jim, we already know about a tremendous diversity of microbes, and there are certainly many more yet to be discovered. And when we talk about microbial diversity, generally we're speaking about genetic diversity, how similar or dissimilar organisms are at the level of DNA. But we also consider metabolic diversity, how organisms make a living. If you think of animals and their energy metabolism, for instance, it's all pretty much the same. Animals eat plants or other animals and then oxidize those organic compounds in the presence of oxygen. That takes place in the mitochondria, and there's really not much variation on that theme. They all do it about the same way. But if you look in the microbial world, there are a tremendous variety of ways that organisms make a living, that is, produce energy to maintain cellular functions and to reproduce.
Now, in an abstract sense, I get what you're saying, but it's sort of hard to picture just sitting here.
Mm-hmm. Well, Jim, let's head over to the lab. We can take a look at some of the microbes and get an idea of how we determine their genetic diversity.
Perfect.
Okay. Jim, to take a look at the diversity of microorganisms, I've selected a termite gut as an example. And the reason for selecting that is in a small volume, we have representatives of all three domains of life, the archaea, the bacteria, and the eukaryotes. So here we have some footage of the microbial life that's inside of a termite gut.
Now, I'm guessing, I'm just a layperson, but I'm guessing these are protozoa, these big ones with the flagellum coming out.
Sure. These are the eukaryotic organisms, the most conspicuous ones in the gut. They're larger, they have a nucleus, and can be defined on those characteristics. In the background, the smaller organisms, here are some spiral-shaped organisms, some cocci and rods. Those are members of the bacteria and the archaea. And there's not nearly the amount of morphological diversity that allows one to characterize those organisms or to assess their genetic diversity.
So if, you know, bacteria and archaea are too small to categorize simply visually by looking at them under a microscope, how do you sort them out?
Right. So what we need to do is to take a look at a part of their genome, and I'll show you how we do that. We'll actually amplify a segment of the genome. This is a gene that encodes for a ribosomal RNA, and that's contained in all life forms, from bacteria to grasses to cows. And we'll actually go through and compare the sequence of those genes, and that'll be our measure of genetic diversity amongst the prokaryotes, the archaea and the bacteria.
Great. And we can do that right here in the lab?
Yeah.
Okay.
Jim, what we have here are some samples of the amplified DNA. What we're going to do here is to load some of the sample onto an agarose gel. We use the agarose just as a means to separate the DNA. So I'm loading into a preformed well here. You see the dye moving down into the well. Okay, so now that we have all the samples loaded, we'll turn on the voltage, and that'll apply a current. The DNA will move through the gel. DNA is negatively charged, and so it'll naturally move towards the positive pole.
Okay, so the current sort of spreads the DNA out for you.
Exactly.
Into a spectrum.
It pulls it based on the size of the DNA. The smaller fragments move faster through the gel than the larger fragments.
Got it.
And we'll then be able to go into the darkroom once the DNA has been stained and visualize that. The next step is to determine the actual sequence of that band of DNA. And we'll determine the sequence and actually compare side by side the sequences that are derived from different organisms.
Okay, and so the gene sequences to a microbe, sort of what my social security number is.
Exactly. It's your social security number or your fingerprint, and by looking at that sequence, we'll be able to identify the organism. We'll know if it's something that we've seen before, or if really it's one of these new organisms that nobody else has ever seen before.
That could be exciting. To fully understand this process, we go to the Institute for Genomic Research in Rockville, Maryland, called TIGER for short. Here, scientists use 60 gene sequencing machines driven by supercomputers to unlock the secrets of DNA from multiple genes. One project currently underway at Tiger is the sequencing of the entire score, called the genome, of Thermatoga, the microbe discovered by German scientist Carl Stetter, living in boiling mud. Dr. Karen Nelson is in charge of the project. Her goal is to determine whether Thermatoga evolved well after life began, or if it is, in fact, one of the closest living relatives of the first life on Earth.
I am driven by being able to answer these questions that we could not answer 5 or 10 years ago with the technology we had 5 or 10 years ago. And now we're sequencing entire genomes in one go, and to me that's just absolutely amazing.
Now, Nelson is part of the greatest explosion ever in our understanding of life's diversity, brought about by the high-speed sequencing of DNA. A key part of the process is attaching 4 different colored dyes, one to each of the 4 different chemicals that make up DNA. It's as if the scientists were trying to decode the score of an incredibly long, complicated song.
I said, woman.
And someone came up with a clever way of attaching a different colored dye to each of the notes. When red dye is splashed on the notes, only the C notes turn red. When green is splashed on the notes, only the A notes turn green. And so on. Then, the color-coded score is loaded into a special, color-sensitive machine that sends the data to a computer. Scientists simply check out in what order the colors are displayed on the computer, and they know in what sequence the notes are being played. It sounds easy, but since scientists need a lot of DNA to work with, they have to start out with many copies of the same score. And since they have to cut all of those copies into thousands of pieces short enough to manage, what the computer spits out in the end is numerous copies of the song in many short segments, completely out of order. A real cacophony. So the hard work in this technique, called shotgun sequencing, is to go over all the pieces again and again and again until you've isolated just one copy of the notes and put them in the correct order.
It takes a lot of dedication, but the completed product is really a reward to see 30,000 sequences going into one single stretch of A, G's, C's, and T's. It's really a remarkable experience.
With Thermatoga's genome sequenced, Nelson and her team began analyzing it and were amazed by what they found. Even though Thermatoga has been classified among the bacteria, it also shares many genes with another branch of life, the archaea.
Although it's a bacteria, it has something that makes it more similar to this other branch of life. And this was a great surprise to us because we didn't think that archaea would have any relationship like this with Thermatoga.
For Nelson, this was convincing evidence that Thermatoga came into being very close to the beginning of life on Earth.
It's evident that Thermatoga belongs at the base of the tree.
The theory that Thermatoga is one of the closest living relatives of the first life on Earth is supported by the fact that every species studied so far, including human beings, has some of the exact same DNA inside as Thermatoga. To see where other organisms fit on the tree of life and how their sequences compare, we can study the organized data on a computer in Dr. Schmidt's lab.
So Jim, we've looked at some of the morphological variability in the microbial world, and we've seen how we can take those microorganisms and retrieve a portion of their genome, and looked at that on the agarose gel. And now what I'd like to show you is what the actual sequence of the ribosomal genes look like. We're looking at the sequence here from a number of organisms, and what strikes you right off is that there are regions where all of the sequences share, all the organisms, all the sequences share the same bases, like over in this region here. There are other regions of the sequence where certain groups of organisms will share a sequence in common, whereas those that border these sequences above and below here, you can see vary in their sequence composition.
So as you determine these variations in the sequences, how does that then translate into how you position these organisms on the tree of life, essentially?
Right. Well, we interpret two sequences that have nearly identical sequence as being closely related. That is, that not much time has passed since they've diverged from their ancestors, whereas sequences that are more divergent, more variable in positions, have had a longer time to accumulate mutations since their common ancestor. And so we look at comparisons of all these sequences and display that result in a phylogenetic tree, which we'll look at next.
Okay.
It appears that life has three primary lines of evolutionary descent. The eukarya, this includes all eukaryotic organisms, plants and animals, but also, for instance, the protist that we saw in the termite gut, and all organisms with a true nucleus. The prokaryotes, though, turn out to be split into two distinct groups, the archaea and the bacteria. The archaea are an interesting group. They contain organisms that live in some of the most extreme environments on Earth. Some of the metabolisms that we see amongst the archaea might be those that are most similar to the ancestors of life, in particularly the capacity to grow at high temperature. That falls amongst this group known as the archaeota. We see here some of the organisms that have been isolated from the deep sea hydrothermal vents. Here are the bacteria. Let's open up this part of the tree. Within the bacteria are a number of groups of organisms. One of the larger ones is the proteobacteria. This is a group of organisms named after the Greek god Proteus, who could change his shape at will. The proteobacteria are capable of immense diversity of different types of metabolism, and frequently can switch from one type of metabolism to another. Within the proteobacteria, we see many of the common organisms, like Escherichia coli from the human gut.
Best known as E. coli, right?
Yep, that's it, the lab rat of microbiologists. Jim, the final group that we might take a look at are the eukaryotes. So
dear to our hearts because we're in that.
We're there.
Yeah.
And here are some common groups of eukaryotes, right? The arthropods, chordates.
That's where we are.
Some of them are medically important, is Giardia and Trichomonas. But there are all sorts of groups of protozoans and amoebae that make up the diversity of eukaryotic life.
Well, Tom, I think I have a good feel for the tree, bacteria, archaea, eukarya, but the one gap here for me is viruses. They're not on here. Why not?
Right. It's a good observation. The viruses currently are not on this phylogenetic tree, nor will they ever be. Viruses don't carry the same complement of genes that we find in other organisms, and so we're unable to construct a phylogeny that includes the rest of life and viruses.
DNA sequencing made it possible for the first time for scientists to systematically explore the invisible world that surrounds us. Now they could ask, who lives here? Who are they related to? And what are they doing? There was just one problem. Scientists could only ask those questions about the very few microbes they had managed to grow in the lab and collect DNA from. Then, Carl Woese's colleague, Norm Pace, made a breakthrough of his own. At its heart was the new technology of cloning, which made it possible for Pace to take a single piece of microbe DNA and make as many copies of it as he needed. Pace realized that he could now collect microbes from any environment and make copies of their DNA without ever having to grow them.
My feeling was that for the very first time, the door to the natural microbial world was open to us. That for the very first time, we could understand what the makeup of the microbial world is, and that is, after all, most of biology.
The first place Pace tried out the new technique was a hot spring in Yellowstone National Park. What Pace discovered staggered even him. Yellowstone's hot springs were home to not just a few otherworldly microbes, but to a stunningly complex ecosystem filled with hundreds of different species. And it isn't just hot springs that are teeming with microbes. It is every environment on Earth. Now, scientists are reading the scores of thousands of previously unknown species of microbes, ushering in a golden age in the study of evolution and of what the tree of life on Earth is really like. Scientists have been surprised not just by how many different kinds of microbes there are, but by discovering that some of those species exist in numbers that rival the number of stars in the universe.
The distribution of life on the planet is very different than we would have thought it even a decade ago. The estimates are that more than 95% of the biomass in the open ocean is microbial. The deep subsurface and the basalts of the crust of the Earth are shot through with life. Life is in ice, life is in waters of boiling temperatures.
But why does it matter that we are surrounded by these tiny living beings? Because it turns out many of them are doing things essential to the survival of life on Earth.
The microbial world is paramount in the context of maintaining the chemistry of the biosphere. The biosphere is fundamentally chemistry. That chemistry is fundamentally driven by microorganisms.
Why should we care? We can't see these things. Well, let's do a thought experiment and take these things away. Let's take all the bacteria in the world away. Do you know what's going to happen? All the life that we can see with our naked eyes, all that life is going to disappear in short order. The microbial world is the basis upon which our whole ecosphere rests. And without it, there is no multicellular life. And a humbling thought is if you do the converse and take away all the animals and plants, you will see big adjustments in the bacterial world, but it will still be there, growing happily along and evolving.
The message of the work of Carl Woese and Norm Pace that microbes play a key role on our planet could hardly be clearer. But what will happen when we can read the entire score, not just of microbes, but of human beings? How will that change our understanding of our role on the planet?
Without microbes in all their diversity, the entire balance of the planet would be in jeopardy. But then it never would have evolved as it did in the first place. And you and I, we wouldn't even be here. Microbes are the very fabric into which all other life is interwoven. And not only can environments influence the nature of microbes, but as we'll learn in our next episode, microbes can influence the nature of their environments.
Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
For information about this and other Annenberg Media programs, call 1-800-LEARNER and visit us at www.learner.org.